So it's a privilege to host once again Hermann Frieboes, who is not visiting us from Louisville, Kentucky, because he's actually still in Louisville, Kentucky, thanks to the wonders of Zoom. So Hermann is an associate professor with two affiliations, one at the Department of Bioengineering at the university, and another one at the Cancer Center, I believe. And he has developed many beautiful mathematical models that incorporate immunotherapy and nanotherapy, has got a master's in computer science from the University of California, Irvine, and also a PhD in mathematical modeling, I think in under the mentorship of John Lovengroup and Vittorio Cristini. So he has established his group at the University of Louisville, where he has really advanced the field of the combination of nanotherapy and immunotherapy. And I'm looking forward to seeing your presentation today, Hermann.
Great, thank you. Thank you so much, David. And thank you for the kind invitation. Last time I was there physically was almost 4 years ago now.
Yes.
And but I'm looking forward to visit you again in person.
Well,
all of
vaccines
you
are coming, I hope.
Yeah. So yeah, and you know, I'm one of your biggest fans. So we're all it's a pleasure for me to give the seminar and to meet you again. So let me switch to my screen here so I can look at my presentation. Okay. So I thought one topic of interest, and this can be very interactive, is to discuss how the immune response changing to a growing tumor is being affected by therapy, in particular by nanotherapy. So the interaction with nanotherapy, the immune system, and the cancer, and then how an integrated experimental mathematical modeling perspective might yield insight into these interactions. And this work is actually being done in collaboration with the Methodist Hospital Research Institute in Houston, with my good collaborator, Dr. Biana Godin. Okay. So as you all know, of course, a lot of these things you actually know better than I do. The tumor microenvironment is very complex with different cell types, different tissues, and in particular with the cancer-associated fibroblasts. We work in the extracellular matrix and other tissues to support the growing tumor. The immune system is involved with myeloid populations, including macrophages, lymphocytes, with an adaptive response, and so on. And it succeeds in many cases in thriving and thwarting the effect of the immune response, so that it will not be essentially eliminated from the body as it tries to survive. And in particular, focusing on macrophages, the monocytes are recruited to the tumor site in addition to already there being tissue-resident macrophages in the tumor as well. And these macrophages, as well as the monocytes, are being affected by the microenvironment in the tumor. And people have studied many of these mechanisms. Here's an example of hypoxia driving the migration of these macrophages and other things. And I think probably a lot of it is still unknown, although a lot of it has been studied from a biological standpoint. In the end, for tumors that succeed, the response that's driven by these macrophages is essentially ends up being favorable to the tumor and not hurting it. And so, and this happens because the macrophage phenotype is flexible, and it can switch from an M1 type, which is on one end of the spectrum pro-inflammatory and cytotoxic, and essentially would promote anti-tumor immunity, to the other end of the spectrum being an M2 phenotype, where it's anti-inflammatory, tissue remodeling, and wound healing. So this phenotype is fluid, and the tumor shifts this towards the M2 type in trying to promote itself, and essentially, not that it's consciously trying to promote it, but it's like a wound that's trying to heal, but it can never heal. And both of these, of course, have their useful purpose in normal tissues. especially in remodeling and growth of tissues, and also, of course, with wound healing. So now looking at nanotherapy, it's been promoted as a means to deliver drugs or other kinds of agents against tumors with enhanced targeting while reducing side effects of these agents. So nanotherapy delivering a chemotherapeutic drug would be touted as being better in terms of being able to focus the drug more on the tumor itself and not on other tissues, such as the digestive system, and also not causing as many side effects because of that. In spite of all the work that's been ongoing with nanotherapy, and for several years now, and probably billions, if not maybe trillions of dollars, only a few formulations have reached widespread clinical application, including liposomal doxorubicin and albumin-bound paclitaxel, known as Abstraction. So one of the big reasons for this is that the complex interactions between the nanoparticles and the heterogeneous tumor microenvironment, including the immune response that's part of this microenvironment, precludes a reliable predictive evaluation of the treatment outcome. And so things that work great with cells in the lab and monolayer, perhaps in three-dimensional spheroid constructs, and then moving on to preclinical mouse with the mouse, and so on, they might work, and maybe perhaps even a few people, but when it comes to the final proof in the pudding, with clinical trials and everything kind of panning out, most of these formulations end up failing. So one big problem is that the highway that's being taken to deliver these nanoparticles and these nanotherapies is the blood vessel, the blood vasculature. And these blood vessels at the tumor site are not normal, which leads to impaired delivery of these blood-borne substances, whether they be even chemotherapeutic drugs as free drug or encapsulated in particles. The vessels can be leaky, so they're not tight. They don't have these tight junctions. The flow can be irregular, sometimes even flowing backwards. They have tortuous shapes that are not anatomically, physiologically proper, and sometimes the vessel distance is too large, leading to areas of tissue that are hypoxic or even almost half dead, but not quite dead. And they're just lying in there, just surviving. And when drug comes in and kills off the ones that are closer to the vessels, these populations can then thrive and reestablish the tumor. So the way I tell my students is like an orchestra. When before the concert we hear the musicians tuning their instruments, and it's all out of whack. All this is really no music. It's just a bunch of sounds, and that's exactly what these vascular endothelial cells are hearing. It's this cacophony of sounds emanating from these tumor cells, and they don't know what to do anymore. One's telling them to go right, one's telling them to go left, one's going to this shape, the other going to the other shape. So whatever they're doing, they're getting very confused and creating this irregular abnormal vasculature, as opposed to a healthy condition where the tumor, the tissue is synchronized and then be able to establish a normal vasculature, which would be when the musicians are playing the concert according to properly. So in these experiments looking at breast cancer nodules in the liver, and this is with my collaborators in Houston. Now, we can actually see that the tumor here on these two upper panels, the tumor itself really has very little vasculature. It's like a dark spot surrounded by the vascularization of the liver. And then the two bottom panels show the normal, the uninvolved liver. So paradoxically, in an organ such as the liver, which is highly vascularized, the nodules that form there from breast cancer, but also from other cancer, including colon cancer, they tend to be very hypovascularized. And part of the reason is because in some way they're feeding off the surroundings, but then they leave the interior very poorly vascularized. And the macrophages that tend to accumulate around these lesions, and we do know that the liver has high numbers of macrophages, they it tends to be on the periphery of these lesions, as we see with the white bars on this chart, compared to the white bars within the lesion on the left or in the uninvolved liver on the right. And I will talk about the loading when the particles come in a little bit later. So macrophages and other phagocytic cells in the body are known to take up nanoparticles, and that's actually how they rid the body of them as well. And in this case, the macrophages in these pictures are shown here on left in blue, and then these particles, these called the multi-stage vehicle particles in green, and they're colocalized, showing that the macrophages have ingested these particles, and this is in the mouse liver. And in three-dimensional cell culture, so with these spheroids are grown with macrophages, and then they're dosed either with nothing, so it's a control, or the paclitaxel by itself, or the paclitaxel encapsulated in the multi-stage vehicle, so in the particle itself. Then, interestingly, the macrophages tend to accumulate more with the encapsulated version. So this graph here shows the growth of these spheroids in the culture. This is the in vitro culture, diameter and sphere area in time over a number of days, and then measuring the cytotoxicity based on free paclitaxel or encapsulated paclitaxel in the particle, or these breast cancer cells in co-culture with macrophages. And the viability was the lowest when it was in co-culture with the particles, and it was actually the worst when it was in co-culture with the free drug, the paclitaxel by itself, as seen here with the triangle indicator. So these are just a few examples of particular conditions being tested. And so when we step back and we think about this, and actually I'm totally preaching to the choir now, because I don't think anybody's going to argue with this, that mathematics is a way as a language to abstract physical concepts for systematic analysis. And it has been applied for centuries in physics to study physical phenomena and other complex interactions, but it's only been applied to biology for about a century and to cancer for a few decades. Mathematics is the language of engineering for the study of complex systems. And now the different types of mathematical models, as you also are well aware, I'm sure. And there's different ways to look at it. And in some ways, they're complementary. And so, for example, statistical models attempt to establish patterns based on observed phenomena, but they might fail to provide insight into the underlying biology because they're more trying to extract the patterns. Mechanistic models represent the known biology, and then very parameters to gain insight into the unknown phenomena. But they can only do so much with what is known in order to project into the unknown. Now, both approaches can be combined. For example, one way would be to use statistical models for determining mechanistic model parameters and the range of values. I know at the IMO at Moffitt, you've done all kinds of work like this, and so that's what's so exciting and so thrilling to actually see the implementation of this kind of thinking in clinical practice. I really believe this is a way to end up helping clinicians be able to cure cancer. I'll give an example of what we applied here in terms of this nanotherapy immune system interactions. So we used a tissue scale model. And so this tissue scale model represents a continuum volume to simulate the transport through the tissue. And so the change in this volume is given by the amount that moves through a unit area per unit time, which is the flux. And there's a source of mass coming from the cell proliferation and a source of and a sink of mass. So mass going away essentially through cell death. So we modeled macrophages as point sources of drug that extravasate from the vasculature in proportion to the local concentration of chemoattractants and, for example, such as pro-angiogenic factors, and to preferentially migrate towards the tissue regions, such as hypoxic tissue, along the increasing gradients of these chemoattractants. So we're kind of trying to mimic what the biology is telling us what's going on. So we have just a simple transport equation for the drug, and then the drug released from the macrophages following these simple kinetics here. And so in this tissue scale model, in a 2D representation, two-dimensional representation, we have on the left two columns, we have the paclitaxel only case, and in the right three columns is when the drug is encapsulated in the nanoparticles. So at the beginning, we have the tumor, and we see the panel here with the tumor with the grid lines, where the tumor itself has a red region, which is proliferating. The blue region is hypoxic, and not much necrosis, mimicking the nodules that are in the liver, the hypovascularized nodules in the liver. And then the grid represents the structure of the liver vasculature, with the squiggly lines, kind of irregular, kind of growing towards the tumor in response to some of this hypoxia, but not quite establishing a full network within the tumor. So at the beginning, when we have the paclitaxel only case, in the second column, first panel, the drug comes in and is at its highest at the beginning. And then over a period of time, it decays. And essentially after 3 days, we see that the tumor has, on the leftmost panel on the bottom, we see that the tumor has slightly shrunk based on this treatment, at least visually, qualitatively. And then on the right three columns.
Herman, can I ask a question?
Yes, yes.
So this is really, really pretty, but I was wondering, given that you've got that kind of lattice that represents the kind of base tissue vasculature, I guess that's what it represents.
Yes, yes.
Does that mean there's normal tissue there sort of utilizing it?
Yes, yes, that's a very good question. I failed to mention that. This is surrounded by the normal tissue, and it is utilizing the vasculature, yes.
So you're not actually showing it, but it's there.
Yes, exactly. Yes, very good question. Yeah, yeah, because otherwise too many things to show. Yeah, and so, yeah, so on the right three columns, when the drug is encapsulated in the particles, then we can, and this is not showing the particles themselves. We actually, in this, we're showing the drug that's eluting from the particles. So the drug is being released. Here's the macrophages. They're actually uptaking these particles in the rightmost column with the white on black panels. And the macrophages actually peak after a certain amount of time. And peak, not the number of the macrophages loaded with the particles releasing drug, it peaks after the bolus case. And what that does essentially, it spreads out the drug over a longer period of time and actually allows the tumor to shrink more than in the case where it was just free in the system. And so then we played around a little bit and looked at, well, what if we did this every 3 days, for example? And then so this upper panel on the left shows a 3-day regimen where the free drug, the free Abstraction is being given in the solid line here, and kind of peaks and decays. And then with the encapsulating the particles, which are then uptaken by the macrophages, which release the drug, then there's a certain delay, as you can see with the dashed lines. And this makes a big difference in terms of the response. On the panel on the upper right, we see that the control is the dashed line. The solid line is the one with the free Abstraction, kind of maintaining control, but as you can see, probably not. And then as it is slightly increasing. And then the case where the bigger dashed line decaying at the bottom is the case where the particles were taken up by the macrophages and then releasing drug over in a more measured way over a longer period of time. On the bottom left here, we calculate the tumor diameter for the control that was not treated, and the Abstraction and the particles carrying the Abstraction, and we have a certain proportion here, seeing that the diameter responded most favorably with the tumor, with the drug that was encapsulated in the particles. And at least here in this case, we compared it proportionally to the in vivo experiments, where there was a control, the free drug, and then the drug encapsulated in the particles, and it was encouraging to see at least a similar trend between the actual in vivo experiments and what the model was doing, giving confidence that at least perhaps we're on the right track here in trying to simulate these phenomena. So with continued experimentation by my collaborators, they noted that the nanotherapy itself affects the macrophage infiltration in the 3D cell culture. And perhaps, hopefully, you can see this, that in the case where the upper colored panel is the control, and the macrophages have their trajectories highlighted in different colors in these squiggly lines. They're kind of random around the spheroid itself, which is on the bottom right corner of the panel with the faint blue rim. And then when the particles come in, there's a certain directionality imparted to these macrophages, but still not totally directional, but there's some directionality imparted. When the Abstraction comes in by itself, there's also directionality given to these macrophages, but they're kind of moving past the tumor, the spheroid. They're kind of moving now, but not necessarily in the direction focusing onto the spheroid. When these two things are combined on the bottom left, we can see the Abstraction encapsulated in these MSV particles. Now they're zooming into the spheroid. The spheroid is barely visible on the lower right of the panel, but they're coming in like arrows into the spheroid, which in itself was very interesting. And they actually quantify these in these bar charts. So it seems that the nanotherapy modulates the accumulation of the macrophages in the tissue. And so on the left side here, when we see the total macrophages, and the x-axis is the distance from the center of the spheroid. So the center is where the y-axis meets the x-axis. And so when the macrophages are loaded with the particles carrying the Abstraction, that's the highest number we see, with the black bar being the highest there and kind of decreasing outwards. And then looking at the actual polarization of these macrophages, interestingly, the M1 macrophages are the anti-tumor and more cytotoxic to the tumor type. They tend to accumulate in higher numbers than the M2 macrophages do towards the center of these spheroids, with the M2 being on the right panel and showing a smaller black bar in the 0 to 50 micron range. Now, it gets a little more complicated because the nanotherapy itself is not only affecting their directionality and their accumulation, it's actually affecting their polarization as well. And so in these pictures, and I apologize, I think some of these images are hard to see, but they're being quantified by these bar charts in red and green colors. So the green being the marker for the M1, the CD80, and the red being the marker for the M2. So when the drug is free, and they actually tried a different drug as well, rapamycin here. When the drug is free, the M1 versus M2 is not necessarily that big of a difference, but when the drug is encapsulated, in this case the Abstraction is encapsulated in the particles, then the macrophages are actually polarizing to the M1 type, as you see by the large green bar here on the left side. And this is with the 3D cell culture in vitro. And a similar effect, not as dramatic as in vitro, was observed in vivo, and that's probably because there's other things going on in vivo. But in the case of the encapsulated drug, again, the M1 macrophages were in higher numbers in this in the tumor here. So we then modeled the effects of the macrophage polarization, and so we separated them into the M1 type, which was simulated to release a cytotoxic substance that would kill the tumor cells, and then the M2 macrophages would release some sort of diffusive growth factor, which would help promote tumor proliferation. And so now with these two, when we look at the simulations, and now the time flows from left to right, and then from top to bottom, 0 to 24, 40, and 72 hours. Again, we have the panel with the tumor in each case, with the grid, the red being the proliferating tissue and the blue being hypoxic. And then the macrophage chemoattractant is shown here, diffusing outwards from the tumor into the surroundings. And then the macrophages are actually white on black, and I have to apologize. These were not the best panels to have here, but the white is these are the macrophages. So they've got the undifferentiated M1 and M2 subtype for at each time point. But the drug that's being released is shown in the upper right for each set of panels, and so we see the same type of effect that the drug being encapsulated in this case, now with the M1s at the same time killing tumor cells, and the M2s promoting the tumor, and then the drug being released from both of them affecting the tumor progression and showing there's some effect from these combined things that are happening here. So we quantify these things. Here we look at the tumor radius and the time after treatment. And so the solid lines here show the case where there's just the drug by itself. And it shows the different types of macrophages in terms of their polarization. And then the broken lines are the cases where the drug was encapsulated. And in all of these cases, the response was better, with the tumor radius decreasing after the one-time treatment. And so we see a case where we totally take out the macrophages. There's no M1 and no M2, or we look at only M1, only M2, or both M1 and M2. And interestingly, we noted that the green dashed line was actually the better case, was actually when both M1 and M2s were present. So killing the tumor at the same time promoting the tumor, as opposed to having M1 only, which was the second best case, only trying to kill the tumor with the M1s. So with this kind of model driven hypothesis that the presence of both would in some way potentiate the effect of the treatment, my collaborators proceeded to try this experimentally and see if this is actually true or not. And so they devised a system with this CRISPR-Richter liposome effect to see if they could actually hardwire the M1 versus M2 polarization, and then in that way manipulate the numbers and see if that made any sense, any difference. So after a lot of work, they were able to fix the macrophages into their phenotype. So even when stimulated to turn to M2, as this bar chart is showing on the lower left, even when stimulated to turn to M2, the M1 phenotype was maintained, as you can see with the large white bar. And so they were quite happy here, and these pictures are actually showing, and you might be able to see just on the bottom set of panels that when they are being stimulated on the bottommost row, third column, there's still quite a number of M1s present, even though they're being stimulated to turn to M2 with the, as you can see with the CD80 marker in red for the M1s. And then using this, they were able to then set up different ratios of M1 to M2 in the 3D spheroid culture, actually co-culture to be precise. And after 48 hours of treatment, we can see that, and these are the ratios at the bottom here, M1 to M2 ratio, 0 to 0, 2000 to 0, meaning all M1s, and then 1500 to 500, 1000 to 1000, 500 to 1500, and then 0 to 2000 being all M2. So the rightmost bar is all M2, and the next to last, the next to the leftmost is all M1s, with the leftmost being a control with no macrophage. So after 48 hours, with no treatment versus with the particles that were loaded with the Abstraction, it seems that having all M1s, having all M2 is actually the worst case. So having all M1s is actually the best case. And then when we looked at a little bit more time, to give it a little more time to see any effects from this, so after 72 hours, then something interesting starts happening in that the case of, you see the white bar here, which is the case of 1500 M1 and 500 M2. So the 3-to-1 ratio here actually elicits a better response than the case where right to the left of it, which would have been all M1s. And we also see some of this happening somewhat on the in the higher ratios. So it's not just increasing, the response is not becoming worse just because the M2s are increasing in number. And so then they went ahead and tried this in vivo, and they were able to hardwire the macrophage phenotype with these breast tumors in the mouse with this CRISPR-Richter liposome treatment. And we can see that with the CD80, the red M1 marker, that the phenotype was maintained in the tumor. And so we looked at this into more detail with the modeling, and we simulate, so to pretend that we were changing the polarization of these macrophages, because they're also changing outside of our control within the simulation as well, based on the local gradients of these chemoattractants and the number of cells and the vascularization, all these other things. So in order to force the phenotype as well, we did this kind of like a hypothetical agent that's injected and drives the polarization in a certain way. So the likelihood of conversion, so the likelihood of the polarization changing, is proportional to the local concentration of this hypothetical agent and its strength. And so here the bar chart shows how in the model, based on the different strengths of this hypothetical agent, we're able to then quote unquote control the phenotype of the macrophages in the simulated tumor with an increasing ratio here towards the M2. So look at the ratio of 3-to-1, which seemed interesting from the previous simulation results. We see here these panels again, and we have them in time from left to right and top to bottom. We have the tumor again on the upper leftmost panels, and then we have the hypothetical immune drug agent coming in through this vasculature and changing the polarization in a certain way, in addition to whatever else is going on here, and the interactions between the tumor, the immune cells, and the particles. And then the drug itself, out of all this, is shown in the upper right panels of each set of panels here. And in this case, actually the macrophages are shown black on white, but it's still not as good to see either, since they're simulated as being point sources of the drug. But we can see that towards the end of the time, the tumor has responded, and as the effect of these different drugs wears out. So in these graphs, in these charts, we actually quantify the what we see just in these pictures. And so in a single treatment, the upper left panel shows the average tumor radius. And now there's a certain stochasticity in here, because the macrophage numbers are variable based on the simulation. It's not necessarily the vascularization, it's a little stochastic. And so we incorporate that as well. And with in the left panel, it shows when M2 macrophages are active, the response is in this way. And the panel B, the rightmost upper right, shows that when the M2s are inactive, so only we have M1s, the response is actually not as favorable. And then when we simulate treatment every 2 days with the bottom left panel, this effect becomes more exacerbated. And we can see that when the M2s are inactive, the response is clearly not as favorable as when both M1 and M2 phenotypes are active. And then we looked at treatment every 3 days, and still, the case with both active was slightly better than when the M2s were completely out of the picture. But interestingly, this also 2 versus 3 days shows how dynamic the response can be based on the timing of these treatments, which needs to be tailored to the tumor conditions in order to eradicate them. And so the presence of this M2 phenotype in some way potentiates the response to the drug that's this Abstraction encapsulated in these particles, and that's being uptaken by the macrophages undergoing M1 to M2 phenotype transitions in the tumor microenvironment. And we can see that when both M1 and M2s are active in this ratio of 3 to 1, then that would give the most favorable response with a treatment of every 2 days here, quantifying what we saw in the previous graph. So one thought is that potentially the M2s being present during the treatment is helping more cells to be sensitive to the treatment, since they're actually making the cells quote unquote happier in their microenvironment, whereas the M1s are making them more unhappy and potentially less susceptible, but at least the M2s are making some of them indirectly be more susceptible to the treatment. And so the efficacy of the nanotherapy depends critically on the tumor microenvironment, in particular the associated stromal cell populations and the macrophages, and as well as other phagocytic cells. And we're looking at other types of immune cells, including T cells, now for a more adaptive response, and how these things could interact with each other and driving each other to promote the tumor. And then how is this affected with the treatment? This polarization of the macrophages in particular is very dynamic and may shift synergistically or antagonistically with the nanotherapy. And so the response to the nanoparticle-mediated chemotherapy targeting poorly perfused tumors, such as these breast cancer lesions in the liver, may benefit from a more fine-tuned M1 to M2 ratio that maintains both phenotypes in the microenvironment during treatment. So perhaps during the treatment, maintaining both, and then in the inter-treatment periods, having only the M1s doing their work. So just because there's some M2s doesn't mean they're bad. And this reminds me of the story with angiogenesis too. And I think you all know the story that angiogenesis is being classified as being bad because it's helping the tumor to grow. But if we don't have angiogenesis, there's not a highway to get to the tumor. There's not a way to deliver things to it. And worse than that, it actually, the tumor cells are alive. They're not waiting to die. They're not just inert objects like chairs and tables. And so if we withdraw the supply of oxygen and nutrients to them, they start moving around looking for them, and the tumor becomes more invasive. And this is like years ago already been proven experimentally. So Complementing experimental investigation with mathematical modeling and computational simulation could help define and test better nanotherapy, targeting these tumor-associated macrophages as well as other stromal cells. And longer term, the computational modeling of immunotherapy might enable systemic evaluation of therapeutics that target patient-specific tumors. And of course, that's what you are all trying to do, not just with immunotherapy, but everything. And that's why I'm, as I said in the beginning, I'm your biggest fan and biggest admirer. Thank you. I'd like to acknowledge my students at the University of Louisville, Louis and Grace, who in particular worked on these simulations, and then my collaborators at the Methodist Hospital Research Institute, Dr. Broden and Dr. Leonard, and then funding by the NIH.
Very cool. Thank you very much, Hermann, for your presentation. It's a beautiful combination of both mathematical model hypothesis-driven research with experiments. It's really you know, like the sort of thing here we at IAMA we like. So do we have any people who might want to ask any questions right now? We have some time.
I have.
Okay. Okay.
So thanks, Hermann, for giving us an update. So that looks like quite an extensive paper that you published in an immunology journal.
Mm.
Was that a difficult process?
Well, it was published in 3 parts because it was kind of a progression of work. And the 3rd part, trying to show that having some M2s would be more beneficial, was probably the more challenging one, but in the end it went through. Yes, it was published in Cancer Immunology, Immunotherapy. Yeah.
Very cool.
Yeah.
So the actual question was, obviously that kind of result is intriguing because, you know, there is a sort of consensus view that M1 macrophages are the ones you want and M2 are the ones you don't want, because M2 sort of facilitate, you know, let's see, they help the tumor, but they also potentially facilitate immune evasion of other immune cell types. And of course, you're not looking at that in your model, but in your experimental system, was that organoids or spheroids? I can't remember. Like,
so the experimental system was spheroids, and also in vivo. Yes. So they have a mouse model.
So there might have been some T cells in the mouse model.
Yes, absolutely. There's things in in vivo that we were not looking at and that need to be looked at. Yes.
But I guess my point here is, what is it about that ratio? And I don't think you quite answered it. I mean, I think you discussed it, but why that specific ratio? Is it because there is this exchange going on between the macrophage state, like there's a switching, and there's something about that ratio that's facilitating a switch? Is that?
So I don't know. I don't think there's anything magic necessarily about the ratio. It's just the ratio, one of the ratios that we were able to achieve experimentally and in the simulations to show a difference between the 2 populations. But I don't think that's necessarily the only ratio that would be beneficial to the therapy. There might be other ratios as well.
Okay.
Yeah, but having some M2s seems to help. At least that I think that would be the conclusion. And they could be explored further to see, yeah.
You know, quickly to that point, are you the first ones to show that, or is there literature that shows this is the M2s and some ratios have a place for favorable outcomes?
You know, we looked at that, and as far as I know, we were the only ones to notice that.
Yeah.
So I'm hoping we're right. But even if you're not right, it's what made me happy is that we were able to drive the experiments based on model results, and that is an achievement in itself. If you know what I mean, you know, I think you appreciate what I'm saying.
I know exactly what you mean. I don't know if immunologists really care about that. That's the point. Right? So they probably care about this other fact, the first one. And so, this is very intriguing. And so, like a roadmap to verify in other tissue types or experiments would be very exciting.
Yeah, and I mean, there's always a philosophy being applied when one does science, right? There's always a philosophy, and so the assumption has been, M1s are good, M2s are bad, and so I don't think they would even.
That's all I knew until today.
Yeah, and it's like the angiogenesis. You know, angiogenesis is bad. No vessels are good, and so it's this assumption that then drives all this experimental work potentially for years and decades, and in the end it ends up not working. And I think in this case, with the model, with the simulations, and I know you guys do this all the time too, is we're able to say, wait a minute, guys, let's look at a different kind of thing from a different angle. Perhaps this or that. It's not necessarily all bad and all good, but there's other things that could potentially also be interesting.
Sandy, had you finished your question, or?
Yeah, I think Philip kind of explored it a little bit further.
Okay.
The only thing that I was going to ask specifically was, in the model, was that ratio something that you saw, right? You know, like, was it just the fact that there was a mix? It wasn't a ratio per se.
Oh, I see what you mean. So we tried different ratios, and that was one of the ones that seemed more favorable. Yeah.
So that did come out of the model, that specific.
Yes. And that's why the collaborators used that ratio in their experiments. Yeah.
Your model, remind me again, is it 2-dimensional or 3-dimensional?
So this one is 2-dimensional.
Okay. Do you think that would make any difference if it was 3-dimensional?
In this case, I don't think it would. That's an excellent question. I don't think it would, because the dynamics are still, I think, would still hold. Yeah.
I agree with
now,
you.
Yeah, I don't know about the ratio. Maybe the ratio is a little different in that case. But that's a very good question. Yeah, we're working on 3-dimensional models too, and that's one of the things we would like to try and see what comes out of that.
Cool.
So hopefully for the next seminar. see how I invite myself.
Maybe in Tampa next time. So Ross, do you have a question? That I feel like you were going to ask something.
Yeah. Herman, really nice talk.
Thank you.
I really enjoyed the integration of the experiment and the model. My question mostly came towards the very end, where you're talking about the clinical implications, which I also liked, is this idea of tumor perfusion. This is something that we really can get a handle on in vivo with imaging, right? So can you talk maybe a little bit more about that idea?
So tumor perfusion in terms of what? In terms of trying to figure out the best nanotherapy, or?
Yeah, I mean, it's meant to be kind of an open-ended question, just because
tumor
perfusion is something I think a lot about, and it's something that we, really can quantify to a very good degree in vivo in humans. So, how do you think about involving that?
Yeah, you kind of hit the nail on the head, right? Because perfusion, it's all about perfusion. I mean, unless we inject tumors directly, which if we're able to get to them that way, might as well take them out in some cases, right? So yeah, perfusion it is difficult to quantify, and it can be tried to be obtained in some way from imaging, maybe with some contrast agents, and then of course with histology, maybe from biopsies or tissue extractions, and seeing, maybe extrapolating from there what we think is in the tumor. But an exhaustive, I would say a more deterministic correlation between, or linkage between, what is seen from biopsies or imaging to what can be simulated in models, I think is a very open area. And because there's really a need for that, so that whatever treatment is being given, whether it's chemotherapy, immunotherapy, nanotherapy in combination of all these things, and has a chance of being more successful based on the particular perfusion that can be measured for any patient.
No, that was all. Thanks, Herman, for a really nice talk. Yeah, I think another thought along those lines would be, could you think of a criteria or a situation in which, you have a threshold of tumor perfusion that would, potentiate a good response, or, a favorable setting for these kinds of therapies, or not, right? Use it as kind of like a biomarker for patients that would be good or less good candidates for these kinds of therapies.
Yeah, that would be fabulous. But I suspect it's probably tied to other conditions within the tumor in addition to perfusion, such as the immune system and the stromal populations, the ECM, and because some tumors like pancreatic tumors are very dense, others are not. And well, of course, with brain tumors, there's all this vascularization going on, but also all this necrosis. So yeah, it's a multidimensional problem. But I think you really hit the nail on the head there. We could have a whole workshop discussing this topic. maybe we should do that in the future.
Right. So just taking it back to the ratio of M1s versus M2s, are you thinking more, and maybe I just missed this in the talk, that this is something that needs to be maintained over time, or is it more about waves of M1s followed by M2s and back to the beginning?
Yeah, I think that doing, at least what this data is kind of telling us, the way I interpret it, is that having some M2s present during the treatment could help to sensitize some of these cells to the treatment. But in between periods of no treatment, it's better not, I would say it's better not to have M2s. Then there's no point anymore. There's no treatment. Let's just have only M1s. So having a manipulation of these populations so that during the treatment, we're getting some indirect, unintended consequence from the M2s being present. Yeah.
Very good. Any more questions?
Well, last time I talked at Muffet, I got a lot of produce thrown at me. I didn't have to go to the supermarket afterwards. At least.
I do not remember that. I don't remember that.
Maybe it was just a dream.
It could not have been more 4 years ago, because I was already here. Maybe
it was
3 years ago.
It was in the spring of 2017.
That's right. I have that in my calendar.
Okay, very early. So I'd just gotten here.
Yeah.
I think I helped you. I helped you move, actually.
Yeah, I remember you holding boxes and.
That guy.
Let me come back to a question, though. What Russ said, I think, resonates a bit, which is you can infer perfusion from imaging, from clinical imaging, right? And so can you then project or make some predictions with your model on a different scale, right? So like your model very much is on a finer scale, but could one take some of those ideas and put it now on like a clinical imaging scale? Have you ever thought about doing that?
Yes. That would be an awesome project. And if anyone wants to work with me on this, I would be very happy. Yes. Yes. I think that would be awesome, Sandy. Yeah.
I'm glad you like it.
We should try to do some of these things together.
Yeah, well, there is a lot of imaging in pancreas. I mean, as you probably know, I mean, these are things that are done both in the research context and routinely clinically. I mean, perfusion and blood flow can really be quantified.
Right.
In vivo. I think Russ is actually signing up for this project.
Well, he's got all these patient images.
Yeah, well, I mean, it's something I think a lot about. Anyway, really nice work, Herman. I like it.
Yeah. Thank you, Russ. Thank you.
Cool. Well, thank you very much, and see you next time here. We'll have the rotten tomatoes and stuff to throw at you.
Or maybe oranges. Florida has good oranges. But
Excellent presentation, Herman. Thank you very much.
Thank you.